Hi, my name is Daryl Khaw. I work in Dr. Virginia Weiss's lab in the Department of Zoology, and my topic for the summer is searching for Trap-6 in Aptasia pallidum. So the purpose of the research in the Weiss lab is to study the symbiotic relationship between corals and their symbionts, which are dinoflagellates or algae. This symbiotic relationship is important because it allows for the formation of the great coral reefs. These reefs not only allow us, you and I, the opportunity to be witnesses of such great beauties that exist on this earth, but these reefs are also the homes and the protection to thousands and thousands of other organisms. And if these reefs cease to exist, so would the lives of these organisms. This topic is of particular interest because coral reefs are currently under the threat of destruction due to coral bleaching. Coral bleaching is when there is no longer that symbiotic relationship between the corals and the dinoflagellate, and is caused by a variety of reasons, mainly human activity, pollution, and climate change. So research must be performed so we can gain insight and learn and understand what we can do to prevent further destruction of these coral reefs. And one particular topic to study is indeed the symbiotic relationship between corals and dinoflagellates. So for my experiments, rather than using coral, I actually use Aptasia pallida, which are sea anemones and are very, very closely related to corals phylogenetically, which make them model organisms for the corals. One of the main reasons why I didn't use corals is because we just simply don't have any coral reefs here in Corvallis. But another reason is that Aptasia, they reproduce at a much faster rate than corals. Aptasia can also be very easily cultured in the labs. They're a lot more versatile than the corals in terms of the environments and the living conditions in which they can survive. Aptasia can survive in environments of different salinities and different water temperatures, whereas corals require a much more specific condition, much more specific environment in which they can live in. Aptasia can also be bleached and kept aposymbiotic. So by placing the corals in an environment that stresses them out, such as cold shocking them 4 degrees in water and keeping them in the dark for a matter of weeks, they begin to spit out the dinoflagellates. And after weeks, they lack the presence of a symbiont, creating them to be aposymbiotic. So aposymbiotic Aptasia is represented by this image here on the right, and symbiotic Aptasia is represented by this image here on the left. Symbiotic Aptasia contains symbionts. So by studying both the symbiotic and aposymbiotic Aptasia, we can gain insight on how the presence of the symbionts affects the lives of the Aptasia, which in turn will give us insight on how the presence of symbionts affect the lives of corals. So the way the symbiotic relationship actually works between the corals, or the host, and the dinoflagellates, are ingested into the host cell, or into the host, and are then absorbed into the host cell, creating this endosymbiotic relationship between the two organisms. The host provides the dinoflagellates with a place to live, a place of protection where they can grow and multiply, and as the dinoflagellates reproduce, they in turn provide the host with the nutrients that they need to survive, thus creating this interdependent relationship between the two organisms. This image here on the left just represents the endosymbiotic relationship between the two organisms, where this is a gastodermal cell of the coral, and these brown spots over here are the dinoflagellates. And this image here on the right just shows the nutrient exchange between the two organisms. So questions are oftentimes asked, why do the host allow the dinoflagellates to live within the cells? In other words, why don't they treat the dinoflagellates as some kind of infection or invader or disease and attack it with the immune system? Which leads me to the next topic, the innate immune response of the host cell. Past research in both the Weiss lab and research centers across the world have provided evidence that the innate immune system of the host is actually involved in the symbiotic relationship between the organisms. This is just a very basic diagram of two of the pathways involved in the innate immune response of the host. Starting with the one on the left is the TLR pathway, the toll-like receptor pathway, which has the purpose of stimulating an immune response. The pathway here on the right is known as the TGF-beta pathway, which has the purpose of suppressing an immune response. So these two pathways, the TLR pathway and the TGF-beta pathway, work hand in hand together to temper and to balance the host immune system. It is further believed that the presence of a symbiont upregulates the TGF-beta pathway and also inhibits the TLR pathway, therefore suppressing the immune response. It is this suppression of the immune response that theoretically allows the dinoflagellates to exist within the cells of the host. So more specifically for my research over the summer, I studied the TLR pathway. Again, this is a very, very basic outline of what the pathway is like, but the idea is that the TLR receptor, located right over here, receives a signal. It then sends the signal down to the next molecule in the pathway, activating that molecule, which forwards the signal to the next molecule in the pathway, creating this downstream cascade in which the end result is the immune response, or for this case, for the TLR pathway, inflammation. The molecule that I studied more specifically for my experiment is known as the Trap-6 protein, located right over here. What's so special and what's so unique about this TRAF6 protein is that most molecules in the pathway are activated through ATP phosphorylation, or through the attachment of adenosine triphosphate, which are energy molecules. TRAF6, on the other hand, is actually activated through ubiquitination, or through the attachment of ubiquitin. Ubiquitin in the past was just seen as a molecule known for, as a marker for protein degradation. But recent research in the past couple years has provided evidence that ubiquitin also serves to activate molecules such as the TRAF6 and signaling pathways, which is one of the main reasons why I'm studying this protein. So the question I was faced with throughout the summer is whether or not the TRAF6 protein is present in aptasia, and if it is, when is it activated? In other words, is the TRAF6 protein only present during immune stimulation, or is it always present but at low concentrations until upregulated by immune stimulation? And I predicted that the TRAF6 is both present and activated in aptasia, but with a greater concentration in immune-stimulated aposymbiotic aptasia. Again, going back to the two pathways, it is believed that the presence of a symbiont upregulates the TGF-beta pathway and inhibits the TLR pathway, therefore suppressing the immune response. Theoretically, it is this suppression of the immune response which should also suppress the presence of the TRAF6 protein. So the immune-stimulating condition that I use for my experiment is through the use of LPS, or lipopolysaccharide, which is the cell wall of a gram-negative bacteria. My experimental group consisted of both symbiotic and aposymbiotic aptasia that were first exposed to filtered seawater and were then exposed to the LPS for 12 hours. The control group consisted of both the aposymbiotic and aposymbiotic aptasia that were just exposed to filtered seawater. And from both the control group and the experimental group, I was able to extract the protein and move on to the next step in the experiment. So using the protein extraction, I was able to perform an immunoprecipitation, which has the purpose of isolating and concentrating the TRAF6 protein. And using that same immunoprecipitated protein, it's able to perform a Western blot analysis, which has the purpose of detecting the size and the presence of the TRAF6 protein. And from the Western blot, I was able to obtain results. So going into a little bit more detail on my method, starting with immunoprecipitation. First, there is the protein extraction, the protein extract from both the control groups and the experimental groups. An antibody specific to the TRAF6 protein is added to the solution. This purple circle right here represents the TRAF6 protein, and this right here represents the TRAF6 antibody. So once the antibody is added to the solution, it then binds to the TRAF6 protein, where the next step is to add special protein G beads, represented by this green rectangle over here. So once the beads are added to the solution, they then bind to the protein antibody complex. And after a few more steps that include centrifugation of the solution and removal of the supernatant, theoretically, all that should be left within the solution are the protein G beads attached to the TRAF6 antibody. And after eluting the beads, all that should be left in the solution now is, again, these purple circles which represent the TRAF6 protein, which are then used in the Western blot analysis. So we have, theoretically, just the TRAF6 protein. They are then loaded onto an SDS polyacrylamide gel for electrophoresis, where an electric current is sent through the gel which separates the protein based on molecular weights. The higher the molecular weight, the higher up on the gel it is. A ladder which is comprised of multiple molecular weights is also loaded onto the gel. And so the purpose of the ladder is so it allows us to detect the size of the TRAF6 protein by comparing the protein to where the ladder is on the gel. So once the gel is complete, the information is then transferred onto a nitrocellulose membrane. But before we can obtain the results, this membrane has to undergo a series of steps, first of which includes blocking. So the membrane is exposed to a blocking solution which has the purpose of reducing the binding between the antibody and the membrane. The membrane is then exposed to the primary antibody, or the TRAF6 antibody, and is then exposed to a secondary antibody, which is specific to the TRAF6 antibody, and serves to label the signal so that the protein can be seen on film. Once the secondary antibody is complete, the membrane is then exposed to film for a certain amount of time, where hopefully the desired result can be seen on the film. So this right here is just an indication of some of the results I obtained over the summer. The lanes are, right here is the positive control, or the HeLa cells, which are known to contain the TRAF6 protein. And then there's the aposymbiotic aptasia, the LPS aposymbiotic aptasia, or the immune-stimulated aposymbiotic aptasia. Then symbiotic aptasia, and then the LPS symbiotic aptasia. So every antibody contains a heavy chain. The heavy chains for the antibodies, the primary and secondary antibodies that I use, are located,